everyone. It's such a pleasure to have Lesley Weaver presenting her work to us today. I'll just give everyone a little bit of her background. So Lesley completed her PhD with Claire Walzak in 2014, studying mitotic spindle assembly at Indiana University. She then switched to flies and started a postdoc at Johns Hopkins in Daniela Drummond-Barbosa's lab, studying how nuclear receptor signaling in adipocytes influences tissue stem cell lineages. And in 2018, Lesley was awarded the really prestigious K99 Transition Award to study how nuclear receptor signaling regulates oogenesis. In 2020, she moved back home to Indiana University and set up her own lab. And what's really special about Lesley's work is what I think is her integrative approach to studying oogenesis and reproductive output. So she's not only just looking at tissue intrinsic signaling, but also the influence of interorgan communication and physiology. And I've been really lucky to know Lesley for over a decade. We first met as students at the physiology course at Woods Hole in 2010. And I guess for the trainees who are in the audience, I really want to emphasize that, you know, your research careers are just as much about the friends you make along the way as the work you do in the lab. And I kind of want to highlight this with a short story of how Lesley's really had a big impact on my career. So about 9 years ago, during a particularly miserable time in my generally miserable PhD, I caught up with Lesley at the annual ASCB meeting. And Lesley was wrapping up. She'd had this amazing PhD. She was moving on to a postdoc. And I remember just like kind of unloading on her about how I hated being a PhD student, how I wished my PhD had ended 5 years before, how I would never find a postdoc, how I didn't know what to do, how I didn't know how to do it, how everything was bad and terrible, and on and on and on. And Lesley was very patient and very kind and kind of reassured me and calmed me down, told me it would all be okay. And then when she got back from the meeting, she sent me the cover letters and her CV and all of the material she put together to be able to apply for postdocs. And it was really nice to see what does this successful application look like. And it really kind of just gave me that confidence boost and the nudge that I needed to get through the next few months. And I've always been immensely grateful. So with that, I will let Lesley take it away, and thank you for making the time to join us virtually.
Well, thank you, Bill, for the very kind introduction. And I can't believe it's been 12 years since we were at Woods Hole. That was such a great experience, I think, for both of us. Like, I think we were both kind of feeling the same, like, imposter-y syndrome. And yeah, so I hope like now when we talk later today, we will flip that and you will give me the encouragement as being an assistant professor. So thank you all for coming to the virtual seminar, and feel free to interrupt me if you have any questions throughout my talk. So adult stem cells are required for tissue homeostasis and repair in response to damage. And so nutrient availability through changes in diet, stress, changes in or aging, and other physiological changes can influence stem cell lineages. And so adult stem cell populations mediate tissue homeostasis, which is intimately linked to the physiology of an organism. So organs are specialized tissues that are composed of adult stem cells that are able to uptake nutrients such as the gut and process them, and then relay these signals to peripheral organs such as the liver or adipose tissue in response to a nutrient stimulus. However, we now know from a lot of work by multiple labs that interorgan communication is very complex and requires coordination between multiple organs. So circulating factors such as steroids, fatty acid derivatives, and metals act through nuclear receptors to modulate the physiology of an organism in response to inputs from the environment. Nuclear receptors are widely expressed transcription factors that are essential for sexual maturation, metabolism, and reproduction. So many studies have focused on how an individual nuclear receptor acts within a specific cell type or tissue to control transcripts that directly will influence that tissue of interest. However, nuclear receptors can act on a variety of tissues, so it's important to think how they may have direct or perhaps indirect functions in other tissues, which at the time I started my postdoc was an underexplored area of research. So as a model, my lab, as a model to understand how physiology can influence reproduction, my lab utilizes the Drosophila ovary, which is shown here. And we do so because not only is it a reproductive tissue, but it is also a stem cell supported tissue. And so here we have a pair of adult ovaries that are connected by this reproductive tract. And at this anterior end, or the translucent end, is where the stem cell lineages are located. So as a cartoon, we can see that each of those ovaries that were shown on the last slide are composed of 16 to 20 subunits called an ovariole. And an ovariole contains progressively older follicles or egg chambers that consist of germ cells that are shown in purple, that are surrounded by somatic follicle cells that are shown in blue. And so each follicle or egg chamber has 16 cells. 15 of these cells will be nurse cells that will support a single oocyte that we can see here as this large cartoon shell. So at the mid stage of oogenesis, the oocyte will begin to grow in size and accumulate yolk proteins through the process of vitellogenesis. And so these mature oocytes will ovulate through the oviduct where fertilization occurs and are then laid on a suitable substrate. So at the anterior end of each ovariole resides the germarium, which houses the stem cell populations that consist of germline stem cells, as well as somatic follicle cell populations. So on average, each germarium contains 2 to 3 germline stem cells that are shown here in dark purple that can be identified based upon their juxtaposition to the niche, which is primarily composed of cap cells shown in pink. Germline stem cells divide asymmetrically to self-renew to generate a germline stem cell, as well as a differentiating cell termed a cystoblast that will undergo 4 synchronous divisions to form an early germline cyst. So in addition to the germline stem cells, there is a population of follicle stem cells, and it's a bit controversial how many follicle stem cells there are in the germaria, but what ultimately happens is that these follicle stem cells will give rise to follicle cells that will surround this developing germline cyst. So there are several features of Drosophila that make it a wonderful model for addressing gaps in knowledge, like how nuclear receptors or other systemic factors can influence physiology. So for one, there are analogous organs that I've highlighted some of them here. There are also many nutrient sensing pathways and components used by mammals like insulin, targeted rapamycin that are also used in flies. In addition, there are many metabolic pathways and enzymes that are conserved from flies to humans. And what's really great about the power of Drosophila is that there are well-established tools for tissue-specific manipulations, which is really important for my studies to understand how inter-organ communication can influence stem cell lineages. So the nuclear receptor superfamily is well conserved from vertebrates to invertebrates, which we can see here. And the Drosophila genome contains 18 nuclear receptors as opposed to 48 in humans, but they each represent each of the major vertebrate receptor subfamilies with little to no gene duplication compared to humans. So many nuclear receptors have been shown to directly regulate specific aspects of oogenesis. For example, HR39 or its human homolog SF1 is required for ovulation, whereas E78, which is the PPAR homolog, is required for early follicle survival. And so what you can notice is that each of these nuclear receptors are acting specifically in the ovary or reproductive tissue itself to regulate oogenesis. So the best characterized or best studied nuclear receptor in Drosophila is the ecdysone receptor, or ECR, which responds to the steroid hormone ecdysone, which is primarily synthesized in the follicle cells of adult females. So work from my postdoc lab and others have shown that ecdysone receptor is required specifically in the germline for regulating the proliferation and maintenance of these germline stem cells, as well as required in a somatic population called escort cells to influence germline cis differentiation. So in addition, it was shown that ecdysone receptor is required in the central nervous system to influence the feeding behavior of adult females, which indirectly influences egg production. And so at the time that I started my postdoc, ecdysone receptor was the only known example of a nuclear receptor acting in a peripheral tissue to influence the behavior of another. So to get back to my question, one objective of my lab is to understand how nuclear receptor signaling in one tissue, such as the adipocytes or muscle, influences the behavior of another. And in this case, I'm interested in the ovary. And I will just apologize in advance. I'm at home and my dog tends to bark at times or groan, which she just did. So if you hear anything in the background, that is her. So to address this question, when I started my postdoc, I did what a lot of postdocs do, and I performed a screen. In this case, it was an RNAi-based screen where I ubiquitously knocked down each nuclear receptor individually in adult females using a ubiquitous Gal4 driver and assayed for egg production as a readout of proper oogenesis. So to do this, I use the Gal4 UAS system, where we have the Gal4 transcription factor under control of the tubulin promoter. So that would allow ubiquitous expression. And then we also combine this with a short hairpin for my nuclear receptor of interest. And when these two flies are combined, the Gal4 transcription factor will bind to this upstream activating sequence and drive expression of this nuclear receptor short hairpin RNA. And so just to show here, I could also have temporal control using an inhibitor of Gal4 called Gal80. That is a temperature sensitive mutant. So I could grow my flies at the permissive temperature of Gal80, where it would inhibit Gal4 activity. And then when the flies were adults, I could shift them to 29 degrees to only allow for expression of Gal4 and my nuclear receptor hairpin of interest, specifically in the adult females. So here we have an example where this transgene is just looking at the expression of a nuclear GFP in different organs of the adult female. And wherever you see green is where this Gal4 is expressed. And so what you can notice is that in the ovary, we have expression in the somatic follicle cell lineages, but there's no expression in the germline. So this allowed for me to have a ubiquitous somatic knockdown of each nuclear receptor of interest. And so what I did is we can have RNAi in every somatic cell and then perform an egg count assay with an egg plate that looks like this. So literally just counting the eggs laid by each female. So from this screen, it was a very successful screen. 13 of the 18 nuclear receptors are required somewhere in the adult female somatic tissue for oogenesis. So one of the nuclear receptors that I chose to focus on is HR4. So on the y-axis, we have the average number of eggs laid per female per day for different time points. And what we can see is that in my control, luciferase control shown in gray, the females lay about 80 eggs at the start of the experiment. But if I knock down HR4 using 3 different RNAi hairpins, we can see that there's a drastic decrease in the number of eggs laid per female. And this is consistent throughout the time course of the experiment. So in mammals, HR4 is known as germ cell nuclear factor, and oocyte knockdown of GCNF reduces fertility. In Drosophila, most of the work has focused on the role of HR4 during the larval stages. And what we do know is that HR4 is required for initiating the timing of ecdysone pulses and feeding behavior in larvae. But the role of HR4 in GCNF and peripheral tissues required to regulate oogenesis was unknown. The decrease in egg production when HR4 is ubiquitously knocked down could reflect a combination of changes or multiple processes that occur in oogenesis, and some of these are listed here. So what I did was I started at the beginning of oogenesis and first assayed how well germline stem cells are being maintained over time in the absence of HR4. So here we have a controlled germaria where the germ cells are labeled in green. The fusum, which is an endoplasmic reticulum-like structure, as well as cap cells or nuclear lamina is labeled in magenta, and DNA is in DAPI. And so I've circled here 2 germline stem cells that are juxtaposed to the niche or the cap cells, which are these bright magenta staining. And so what I did is I can count the number of germline stem cells over time, and we can see that there's a slight decrease in germline stem cell number over time, and we know that that is due to aging. However, when we knock down HR4 in all of the somatic tissues of the adult female, what we can see is that there's a drastic decrease or increase in the rate at which germline stem cells are being lost. So what we're looking at is we're looking at the change or the slope over time, and we can see that the slope is a lot steeper in these blue lines compared to the gray control. Furthermore, if we just look at the germaria, it looks drastically different compared to control. It's shorter in size. We can see that there's one germline stem cell here that's remaining at the niche, but there are a lot of these differentiating cells or cysts that are missing from the HR4 knockout. And so this could occur due to a number of reasons, or a couple of reasons. One, this decrease in differentiating cysts could be because the germ cells or germline stem cells that are remaining are not proliferating as much compared to control. Or the second reason could be that these cysts are dying after they begin differentiating. Oh, and then I forgot to mention that in this case, like, it may not look like there's a big difference, but remember that each ovary contains 16 to 20 ovarioles, and then there are 2 ovaries per female. So over the course of 14 days, these females are losing up to 90 stem cells. So it's quite dramatic. Okay, so I next looked at how well germline stem cells were proliferating when HR4 is knocked down in all the somatic cells of the adult female. So here we have 2 examples of germline stem cells. In this top panel, we have a germline stem cell that is outlined and is labeled with the S phase thymidine analog EDU. And in control germline stem cells, there are about 6% of the germline stem cells are in S phase. And then we could also stain for mitotic germline stem cells using phosphohistone H3. And in control, about 3% of the germline stem cells are in metaphase or in mitosis. However, when HR4 is ubiquitously knocked down, the percentage of germline stem cells in both S phase and M phase are significantly decreased. And so this indicates that HR4 is required in some somatic tissue or cell type for regulating germline stem cell proliferation. So I next assayed how well germline cysts are surviving in the germaria after they begin differentiating. And so to test this, I labeled the germaria with APOP tag, which recognizes double-stranded breaks in dying cells, which we can see here. And in control knockdowns, about 20% of the germaria have apoptotic cysts. And when HR4 is knocked down, we can see that the percentage of apoptotic cysts drastically increased. And so at least twice as many germaria contain dying early germline cysts compared to control. So we've seen here that loss of HR4 in all the somatic tissues of the adult female influences germline stem cell maintenance and its proliferation, as well as how well these early germline cysts survive. So changes in physiology can also influence the growth of follicles and their later survival during vitellogenesis. And so I therefore tested whether HR4 in somatic cells influences the growth of these growing egg chambers, as well as entering this process of vitellogenesis. So again, I took advantage of the S phase analog, thymidine analog EDU, as well as phosphohistone 3 to label the follicle epithelium where the cells are in S phase or M phase. And so under control conditions, about 25% of the mitotic follicles are in S phase, and about 5% of those cells are in mitosis. However, when HR4 is knocked down, we can see that there's a dramatic decrease in the number of follicle cells that are in S phase or M phase. So this suggests that HR4 is required to regulate the growth of the developing egg chambers. So how well do egg chambers undergoing vitellogenesis survive when HR4 is knocked down? So again, we have a control ovariole shown here, and we're just looking at the DAPI staining. And what I've highlighted are 3 vitellogenic follicles. So what we can see are the nerve cells here that are these brightly colored DNA cells, and then the developing oocyte, which has started the process of vitellogenesis here. And so in a control ovariole, we can see that there are rarely any dying vitellogenic egg chambers. However, when HR4 is knocked down, what we can notice is that these nerve cells that are pretty round and happy in the control have become pycnotic. And so there's high puncta in these cells, and that's an indicator of death. And so compared to the control, we can see that when HR4 is knocked down, about 30% of the ovarioles that have been analyzed have death of vitellogenic egg chambers. So in the data that I just showed, HR4 is knocked down in all the tissues of the adult female. So now the question becomes, what tissue or tissues require HR4 to influence these aspects of oogenesis that I've highlighted here? And so it's unclear from the global knockdown whether or not HR4 is required in multiple tissues or one specific tissue for each of these aspects of oogenesis. So to determine what tissues may require HR4 activity to regulate these processes of oogenesis, I just examined where HR4 transcripts were located in the adult female, looking at the whole fly, ovary, gut, the abdominal carcass, which is similar to the fat tissue, fat and liver tissue, the thorax, which is similar to the muscle, and the head. And so what we can see is that HR4 transcripts are enriched in both the ovary and the muscle. And so muscles have recently emerged as an endocrine organ to help maintain energy homeostasis through the secretion of myokines. So this has gained special interest of lately how muscles are secreting these myokines to influence different physiological processes. So healthy muscle has been implicated in preventing pathophysiology of diseases. Myokines, or these muscle-derived endocrine factors, such as myostatin and interleukin-6, can signal to local and distant tissues to control tissue function. There have been some recent reports that muscle signals can influence brain activity. And it's predicted that there are over 200 secreted myokines, and the majority of proteins are yet to be characterized. So I assayed all of the processes. So I wondered if HR4 was regulating oogenesis from the muscle. And so I performed analysis of all the processes that I described before, where I knocked down HR4 specifically in the muscle using a muscle-specific driver. So I'm just going to go through the processes that were affected when HR4 was lost specifically in the muscle. And so what we can see here is that if we look at the average number of germline stem cells over time, with the luciferase control shown in gray, knockdown of HR4 specifically in the adult female muscle increases the rate at which germline stem cells are being lost from the niche. In addition, I found that HR4 knockdown is required for proliferation of the mitotic follicle cells, which is an indicator of how well egg chambers are growing. And so here we have the percentage of S-phase follicle cells or mitotic follicle cells for the control. And when we knock down HR4, we can see that the percentage of cells in these two phases are significantly decreased. So this suggests that HR4 is required specifically in the adult female muscle to influence germline stem cell maintenance, as well as follicle cell proliferation or egg chamber growth. So as a reminder, there was an abundance of HR4 transcripts in the ovary. And so I hypothesized that HR4 could be acting in the germline stem cell niche to regulate either maintenance or proliferation of the germline stem cells. It could be acting in the germline itself to regulate these processes, as well as progeny growth, differentiation, or survival. And then there is a population of escort cells, which promotes survival of the differentiating progeny. So it could be possible that HR4 was acting in these cells. So I first wanted to address whether HR4 is required specifically in the germline, because its homolog GCNF, knockdown of it will reduce oocyte quality. So to do this, I did mosaic clonal analysis. And so what we can do is generate these mosaic clones in the context of heterozygous organisms that contain flip-based recognition targets, or FRTs, on the base of specific chromosome arms. So on one chromosome arm, we have a mutation of our gene of interest, and in this case, it's HR4. And on its homolog, it has a wild type allele of our gene of interest and also carries a marker. And in this case, it's a GFP marker. So under control of a heat shock flip base, we can induce mitotic recombination and allow for switching of these two regions of the chromosome arms where the FRT is located. And so based on separation, random separation, you could have an outcome where you have two GFP positive heterozygous cells, which would just appear as green. Or in some cases, you can have a twin spot where based on segregation, you have a GFP negative mutant cell that's shown here, juxtaposed to a GFP positive wild type cell. So what does this look like in the ovarian tissue? So here we have a control germ area in which mosaic clones have been generated. And I just want to note that in this case, both GFP positive and GFP negative cells have two wild type copies of HR4 alleles. And so what we can see is that we have this GFP negative germline stem cell shown here next to a GFP positive germline stem cell. And what we can look at is how well their progeny are surviving. So we can see that there is GFP negative progeny that correlates back to this GFP negative germline stem cell. And so in this case, where we have an HR4 mutant allele, we have two GFP positive germline stem cells, that one is shown here and the other is shown here. But what we can realize is that there is GFP negative progeny within the germ area that cannot correlate to a GFP negative germline stem cell. So this indicates that this germline stem cell that gave rise to this progeny has been lost from the niche. And so we can quantify the percentage of germline stem cell loss events here. So this does occur in control cells about 6 to 7% of the time. But when we have HR4 mutant alleles, we can see that this has dramatically increased. And so this suggests that HR4 is required in the germline itself for germline stem cell maintenance. So I found in the previous experiment that HR4 is similar to its homolog and required cell autonomously in the germline for proper oogenesis. So I next wanted to determine whether the somatic populations of the ovary required for HR4 were required for proper oogenesis. So in results that I'm not going to show today, I found that HR4 is not required in the somatic population of escort cells to influence progeny growth or survival. So I next looked at whether HR4 is required in the niche for proper germline stem cell maintenance or proliferation. Oh, and I think I took those slides out. But what I found is that HR4 is required in the niche for germline stem cell proliferation. So I did a similar assay where I looked at how many, or the percentage of germline stem cells that were either in S phase or M phase when HR4 is knocked out specifically in the CAP cells. And what I found is that loss of HR4 specifically in the CAP cells did decrease the number or percentage of cells in S or M phase, but it did not have an obvious effect on how well germline stem cells remain maintained in the niche. So to summarize this part, what I found is that HR4 is required specifically in the adult female muscle for germline stem cell maintenance, as well as how well these egg chambers will grow. And I also found that HR4 is required specifically in the germline itself for germline stem cell maintenance, similar to its homolog GCNF, and HR4 is required in the niche for germline stem cell proliferation. And so it's still unclear at this time. Me and people in the lab are trying to uncover what are the factors that are downstream of HR4 in either the muscle or these ovarian cell types that are regulating these distinct processes. In addition, from my work that I have shown so far, there are 2 additional phenotypes that cannot be, that were not sorted out as far as where HR4 is required for these processes, including early germline cyst survival, as well as vitellogenic follicle survival. So hopefully I will recruit a student this year that will begin dissecting the requirement of HR4 in the central nervous system. There's some evidence that HR4 may be required in adipokinetic hormone producing cells in the brain, as well as insulin producing cells in the brain. So that will be really interesting. We don't know if HR4 is required in the fat cells, so the adipocytes or the enocytes, which are kind of, the enocytes are liver-like cells in Drosophila, or in the many populations of the gut. And of course, we would then need to know what the downstream factors are of HR4 in these tissues. So one way that we can begin addressing some of these questions is understanding how is HR4 activity in muscle regulating oogenesis or any specific cell type. So the easiest explanation, or the simplest explanation, is that HR4 is required in the muscle. It regulates some secreted transcription of a secreted factor that directly goes to the ovary. But it is a little bit more complicated. It could be that HR4 is influencing some signaling pathway in the muscle that then indirectly influences secreted factors that go to the ovary. And I always get in trouble when I present this because it's very linear, this is what's happening. But I just want to be transparent and say that it's actually even more complicated, because these secreted factors could then go on to some other tissues, such as the brain or the fat tissue, which would then indirectly signal to the ovary. So I'm just being very transparent and saying I don't think it's always a direct muscle to ovary communication, even though in some cases it could be that way. So that just means that we have a lot of work to do in my lab. But how can we begin to address some of these questions? So one way is that we can look at or identify HR4 targets. And so what we can do is we can knock down HR4 in the muscle, isolate the muscle, and then perform RNA sequencing, where we look for transcripts that are either more abundant or less abundant in the transcriptome when HR4 is knocked down. And so we would predict that maybe we would have secreted proteins, signaling molecules, or even metabolic pathways that would be enriched. And so you can do what a lot of people do is do PCR validation and then do an RNAi screen with muscle drivers. So that's one project that we're currently working on. But how do we get to whether or not, so this would influence whether or not HR4 is directly regulating transcripts in this sort of way. But how can we say what secreted factors are directly coming from HR4 in the muscle and going directly to the ovary? And so about a year ago, the Perriman lab created this platform to identify secreted protein trafficking between organs using an engineered biotin ligase that will promiscuously biotinylate proteins in a subcellular compartment of the cell. And so we have constructs that will either be targeted to the endoplasmic reticulum or our cytoplasmic. So biotinylated proteins, so we can feed the flies biotin in the food. And so this will allow for any proteins that are to be secreted to be biotinylated. And so this is representing the hemolymph, but we can enrich for biotinylated proteins in our target of interest, which is the ovary, and identify them using mass spec. So we would crush up the ovary, have a mixture of biotinylated proteins and untagged proteins, pull down these biotinylated proteins using streptavidin beads, and then we can purify them and identify them based on mass spec. So that's something that we're trying to optimize right now in the lab. It does take a lot of ovaries. So I think we found that our optimum is dissecting 600 to 800 ovaries to get a good read. And so we did a test run of this using adipocytes where we have known factors like yolk protein 1 and 2 and lipofluorin that we were able to recognize on the mass spec. So it's something that is in progress. So I can pause here before moving on if anyone has any questions about HR4 in the muscle. Okay, well, I'll move on to tell about another story that I worked on. So when I joined Daniela's lab, they had previously found that many pathways in the adipocytes, including the amino acid response pathway, as well as insulin signaling, have a role in regulating germline stem cell maintenance. But the question became is what are these secreted factors coming from the adipocytes that are able to influence this process of oogenesis? So in an effort to identify some of these factors, the German Barbosa lab compared fat bodies from females that were fed a rich diet of protein, yeast protein, versus a poor diet that was composed of molasses. And so they just fed the flies for 12 hours, dissected the fat bodies, and then performed eye-track analysis or quantitative proteomics approach to identify proteins that were more or less abundant in the fat when the flies were switched to this nutrient poor diet. And so what they found was that about 226 proteins were more abundant in the fat on a poor diet, where 50 of those were predicted secreted proteins. And then about 220 of those were downregulated or less abundant with 19 of those enriched. And so what was interesting that when we looked at the data set, we noticed that all of the components of the Drosophila extracellular matrix are more abundant on the fat in a poor diet. So what we have on the y-axis is the poor to rich ratio, and anything that is greater than 1 means that it's more abundant in the fat tissue when the flies are fed a nutrient poor diet. And so what was interesting was that components of the extracellular matrix collagen 4 complex was more abundant in the fat when flies were fed a nutrient-rich diet. And so I wanted to ask how well was the requirement of collagen 4 in the fat tissue in regulating oogenesis. And so what I did was I used an adipocyte-specific driver, and I knocked down either subunit of collagen 4, which in this case implies it's CG25C or Viking. And so what we can see, if we look at these graphs that I've shown before, knockdown of either subunit of collagen 4 significantly decreased the number of germline stem cells laid by the females over time. And so this suggests that adipocyte-derived collagen 4 is required for oogenesis. So we wondered how collagen in adipocytes might control the number of germline stem cells. So if you look at a protein reporter of collagen 4, which in this case it's Viking fused to GFP, we can see that collagen surrounds the whole germaria. So all of these egg chambers are surrounded by collagen. But what was interesting is that if you look at a transcript reporter for CG-Gal4, just looking at a nuclear GFP, we can see that collagen transcripts are only detected in the early stages, that there's no collagen 4 detected in the early stages of oogenesis, and it's only when the egg chambers begin this process of vitellogenesis that you can see collagen 4 transcripts being made. So we hypothesized that collagen 4 is being made in the adipocytes and secreted through the hemolymph, where it could incorporate into the extracellular matrix of the germaria to influence stem cell progeny. However, it was unknown at this time whether or not collagen 4 could be secreted and incorporate into the extracellular matrix or the basement membrane of a fully established adult tissue. So to assay for this, I generated a fly that contained 1 copy of collagen 4 or Viking GFP, and then 1 wild type copy. And so that way we could get rid of the GFP Viking so that the flies would survive. And so I knocked down the GFP Viking or collagen 4 all throughout development. And once the flies became adults, I could then either keep GFP Viking turned off or turn it back on by shifting the flies to that Gal 80 permissive temperature to allow GFP Viking to be turned back on specifically in the adipocytes. And then I could assay what is happening in the germaria. So as a control, we're looking at GFP Viking levels around the adipocytes, where we're doing all of these manipulations to make sure the system is behaving as expected. So when GFP Viking has been turned off throughout development, we can see in these 0-day-old adult flies that there's hardly any collagen detected around the adipocytes. And the quantification is shown here. If we keep GFP Viking turned off during adulthood for 21 days, we can still see that there's very low levels of that GFP detection around the adipocytes. And what was nice is that if we allowed for GFP Viking to be turned back on, we can see that there is nice GFP collagen located around the adipocytes. So we can now that we know that our controls are working, we can next ask what is happening in the ovary when these GFP Viking levels are manipulated. So here we have where GFP Viking has been turned off during development in the adipocytes, and we don't see high levels of collagen 4 around this stem cell area. And so that was what we predicted based on previous reports that larval adipocytes secrete collagen that is incorporated into developing organs. And if we can keep the GFP Viking turned off during adulthood, we still see that there's very low levels of collagen 4 around this germline stem cell area of the germarium. But what was interesting and surprising is that if we allow GFP Viking to be turned back on, specifically in the adults, we see this increased enrichment of collagen 4 around this germline stem cell niche area. So this suggested that I found that adult adipocytes can secrete collagen 4, specifically during adulthood, that travels through the hemolymph and is incorporated into the basement membrane of the germaria. And to our knowledge, this was the first time showing that the extracellular matrix proteins from another tissue could be incorporated into a fully established basement membrane. And so, and to influence germline stem cell behavior. And so the question becomes, does adipocyte collagen regulate known local signals in the germaria that can influence germline stem cell number? So the two main pathways that are present that are important for germline stem cell maintenance are the BMP signaling pathway, where BMP ligands are secreted from the CAP cells and incorporated in the germline stem cell to maintain its identity, as well as e-cadherin levels that are juxtaposed at the niche, which helps glue the germline stem cells to the niche cells. And so it's been shown that collagen 4 signaling can influence both of these signaling pathways. And in results that I'm not going to show today, I found that BMP signaling is not affected by adipocyte knockdown of collagen 4. So I next tested whether adipocyte collagen 4 influences e-cadherin levels at the niche. So here we have the control germaria where we have e-cadherin labeled here in white or in red, which we can see here. And there are two germline stem cells outlined here. And so when I looked at the total e-cadherin intensity in the CAP cells, we can see that quantification here. However, when Viking or collagen 4 is knocked down specifically in the adipocytes, there's a significant decrease in e-cadherin levels shown here and in the gray image here. And so this suggests that the germline stem cells are lost due to a reduction in adhesion when collagen 4 is knocked down specifically in the adipocytes. So my model that we left this paper with is that collagen 4 is secreted from adult adipocytes and incorporated into the germarium extracellular matrix to regulate germline stem cell number, specifically by promoting adherence to the niche. And in support of this model, I found that adipocyte collagen 4 also interacts with known regulators of e-cadherin levels, including beta integrin and focal adhesion kinase to influence germline stem cell number. So in summary, what I've shown you today is that I found that HR4 is required in adult female muscle to regulate germline stem cell number as well as follicle growth. And we're still trying to figure out what are these downstream targets of HR4 in this specific tissue to regulate these processes of oogenesis. In addition, I showed you that we do know of one secreted factor from one tissue, such as collagen 4 or other extracellular matrix proteins as well, can influence focal adhesion kinase to regulate the levels of e-cadherin to influence germline stem cell number from the adipocytes. And in work from my postdoc that I didn't show is that I found that another nuclear receptor called 7up acts specifically in the adipocytes to influence germline stem cell number as well as early cyst survival, and then works is required in the hepatocyte like enocytes to regulate the process of vitellogenesis. And so from some RNA sequencing work, we believe that 7F is regulating amino acid storage proteins, as well as detoxification factors to influence these aspects of oogenesis. And so I'd just like to also leave you with this quote that I found shortly before I began my my assistant professor position, and that the hormone system works like a democracy. Every tissue in the body is an endocrine organ asserting its needs and demands. And so really every tissue in our body is communicating to the other to say, you know, do I have enough energy? Are there enough nutrients? And if there isn't enough nutrients or energy, there has to be a consequence. And in the adult female of Drosophila, usually oogenesis is the sacrifice, because it takes a lot of energy and nutrients to produce progeny. And so my work has focused on how 7Up or nuclear receptors, such as HR4, act in specific tissues to regulate processes of oogenesis. But it's also important to remember that flies or humans do not live in a vacuum. So it will be interesting to understand how the signaling downstream of nuclear receptors can be influenced due to systemic changes, such as change in age or exposure to different diets, and how that may affect the response in oogenesis or other stem cell lineages in the body. And so I'd just like to thank my postdoc lab, specifically Daniela, who took me on. Like, I guess another note to trainees is don't be afraid to try something new. I went from cell culture and xenopus during grad school to fruit flies during my postdoc. And it was another learning curve for me, but it was sped up because I had all of the training from my grad lab. Sources of stocks, reagents, funding. I currently have 2 people on my lab, so a research associate and 1 grad student, and then 2 undergrad volunteers. And with that, I'd be happy to take any additional questions.
Thanks, Lesley. That was lovely. Any questions? Roberto?
Yes, nice talk. I have 2 questions. One is when you did the manipulation of collagen in the adipocytes, then you observe an effect in the ovary in the position of collagen. Did you look at other organs to see if this was a general effect or was a specific effect on the ovary? That is one.
Yeah, that's a great question, and I did not do that. I think that would be great to have an undergrad do, because I still have all of those stocks, and it's a pretty easy experiment to do. Yeah, so no, I haven't done that.
The other question is something that was not very clear to me, is that I mean, you show that this lack of collagen in the ovary, it seemed to affect the level of E-cadherin, but it was not clear to me if you know if this is at the transcriptional level or at it's just the localization of E-cadherin that is different. And the reason why I'm asking this is because there is good evidence, mainly from in vitro studies, that there is as you say, there is a crosstalk between integrins and E-cadherin regulation, but goes in the opposite direction, in the sense that if you have more FAC or integrins, usually you have less cell addition. So that is why I was trying to understand that.
Yeah, so yeah, it is opposite. I don't know if it's at the level of transcription. All I did was look at the protein levels, but and results that I didn't show is if I get rid of focal adhesion protein in the niche, that also decreases the E-cadherin levels. So I think it's opposite to what is shown in cell culture for sure.
Yeah, thank you.
Mark is next on my screen.
Hi, thanks. That was a really nice talk. I had a question about the HR4 part, where I guess So I don't know if I missed this, but is it known what activates HR4? Is it just constitutively active? Or is it something that's going to change depending on the environment? I guess you kind of alluded to that at the end, like that, there could be some environmental factor that leads to activation of all of these things. And do you have an idea what that could be?
I don't. So HR4 is an orphan nuclear receptor. You didn't miss it. And that's a great question. We don't know what its ligand is or if it has one. So I think one advantage of being in Bloomington is that the DGRC is on the same floor as me. So we can do some screens and cell culture just to see, like with a luciferase reporter, what could be activating HR4 or any of these nuclear receptors of interest. So yeah, we don't know what its ligand is right now.
Yeah, along the same lines, actually, I was going to ask if there was, because I just don't know much about nuclear hormone receptors, but
Okay.
if there was a transcriptional reporter you could use to study activation in vivo in response to nutrients and or infection or so on.
We can try and make one. Right now, there's a ligand binding reporter that has been made. There are fly stalks as well as cell like DNA plasmids that we can use for cell culture analysis. But yeah, that could be something that we'd look into, trying to make a transcriptional reporter, or just like knock in GAL4 at the HR4 locus and see what's turned on or not. Yeah.
I think Anna's next.
Yeah. Hi, very nice talk. I also had a question about the fat body extracellular matrix access to the egg chamber. Do you know whether it's a more local, I don't know, do you think it's a fat body, that the part of the fat body that is closer to the ovary that does this job basically? And have you tried to sort of, is there some way of basically blocking the collagen secretion in the fat body, but bringing it back from different parts inside the body in other cell types? You're overexpressing it. I don't know, maybe that doesn't work.
So I don't know what part of the fat. So there's like head fat. So I didn't show a picture, but there's fat everywhere in the body of the adult female. Stop it, Zoe. Sorry. And what recently has come out is the single cell RNA transcriptome of the adult fly. And so there are ways that maybe there is like specific head fat targets versus body transcript levels that maybe we could begin to tease up apart, like what part of the fat it's coming from. You can, it would take some clever genetics, but there are ways that I could probably knock out the fat or knock out collagen 4 in the fat and then try and overexpress it or add it back from another tissue. But I would have to think about how to do that a little bit more genetically. Yeah.
Sure. Okay, thanks.
Or I guess one way is that you could use, you can't get rid of it everywhere because the flies will die. But I was thinking if you could like have a global mutant and then express in specific tissues to see where that could be important. Yeah,
thanks.
Sorry, did you say me?
Yep.
Oh, great. I just had a little squeak. Sorry. Thanks, Lesley. It's Chris here. It's a fantastic talk. Thank you very much. I wanted to ask a question about the HR4 knockdowns in the muscles lineage. Do you have any idea what the metabolic phenotype of those animals are once it's knocked down, and what's their kind of behavior like in that condition? Are they still mobile? Are they moving around?
So they are still mobile. Were you my reviewer for this paper? Because that was a question. They are mobile. They seem to fly just fine. I haven't looked at any type of feeding behavior or any other types of behaviors. So as far as I can tell, they seem to be okay when you knock down HR4 in the muscle. And I missed the first part what type of phenotypes
Well,
were you saying?
what's the kind of metabolic phenotype? I mean, are they active?
Oh, metabolic.
So if you
Yeah.
were, for example, if you were to put in some kind of agent to paralyze the muscle, would you get similar types of effects on oogenesis?
You know, that's a good question. And so the other thing to take into account is that there's a muscle sheath surrounding the ovary that allows it to contract to release the eggs. I've been asked, like, do I think that this muscle knockdown is affecting the muscle sheath? I don't think so, because the driver I used isn't well expressed in the muscle. But as far as other, or of the ovarian sheath, but other metabolic phenotypes, I didn't look carefully. So this is definitely a pandemic paper that I got rid of experiments during my postdoc, and then was just trying to finish when I started my lab. But I didn't look at like levels of lipids in the muscle or the fat. If I just looked at the muscle itself when HR4 is knocked down, there weren't any gross morphological defects in the muscle. So
Okay.
it seems like the flies are generally okay.
Thank you.
Mm-hmm.
I have one question. It's sort of along the lines of, I think, what Mark was asking you. So the fact that you see HR4 having, you know, specific effects in specific cell types in the germline, but also in the muscle, and then potentially from other tissues. I guess, is there any evidence that HR4 is nutrition dependent, or its activity nutrition dependent? Are you able to sort of express an activated form of the receptor under conditions of starvation or something like that to see a rescue?
Yeah, so we do know from the proteomics study that Daniela's lab did that in response to a nutrient poor diet or a high sugar diet, so to speak, HR4 protein levels are increased on a nutrient poor diet, at least in the fat. So it does seem like there are some nutritional cues. I would hypothesize that it is like in response to its activity is in response to nutritional cue, because in larvae, HR4 is a regulator of ecdysone signaling, and ecdysone is sensitive to changes in nutrition. But as far as what specifically, I don't know. A lot of the nuclear receptors are orphans. We do know, like, HNF4, hepatocyte nuclear factor 4, it is responsive to certain lipids. There's a nuclear receptor in Drosophila called E75 that can respond to gas or heme, but it's kind of bare bones what we know so far.
Cool, lots to do.
Yeah.
Okay, with that, thank you so much.